The overall goal of this experiment is to characterize chemical composition of exhaled breath using secondary nanoelectrospray ionization coupled to high-resolution mass spectrometry.
This method can help to answer key questions in the medical field, such as early diagnosis of diseases, therapeutic drug monitoring, and environmental exposure. The main advantage of the technique is the measurement can be performed in real time. The technique is also high throughput, sensitive, specific, and user-friendly.
The implications of this technique have been extended to the diagnosis of lung cancer because tens of biomarker candidates have been found in exhaled breaths of lung cancer patients at different stages.
Generally, individuals new to this method will struggle because the ionization source is commonly homemade. Visual demonstration is critical, as there is no standard protocol for this method yet.
To begin, set up a secondary nano-ESI source according to the secondary electrospray ionization process, in which breath gas is introduced to intersect an electrospray plume and ionized by the charged droplets.
The sources built in individual labs depend on the mass spectrometer used. Here, the secondary nano-ESI source is based on a commercial nano-ESI source and attached to a benchtop quadruple orbitrap mass spectrometer.
The main body of the source is a cubic stainless steel chamber with an inlet to introduce the nano-ESI capillary into the chamber. 2 stainless steel tubes are installed on each side of the chamber for gas delivery.